Thank you for your interest in our research. My name is Wei Wei. It is my pleasure to present our research today. In this poster presentation, I will talk about our research on applying UniFrac to whole genome shotgun data. Let's begin by understanding a little background on this study. First, what is UniFrac? UniFrac is a metric that is widely applied in measuring the diversity between two metagenomic samples, what is also known as beta diversity. Traditionally, it is only applied on 16S rRNA data, based on which we can construct a phylogenetic tree. A phylogenetic tree shows the hierarchical relationship between the organisms present in the two samples and the evolutionary distances among the organisms, represented as branch lengths. The UniFrac is computed based on these two pieces of information. In our research, we made a novel attempt to study the possibility of applying the UniFrac to whole genome shotgun data, which is better than the 16S data in terms of accuracy and resolution. However, unlike 16S data, it is almost impossible to construct a phylogenetic tree using WGS data. However, we can produce a taxonomic tree which is similar to a phylogenetic tree in structure, with only the absence of branch lengths. We developed a workflow to compute UniFrac using taxonomic profiles from whole genome shotgun data. We call this extension WGS UniFrac. This is a workflow of our method. First, whole genome shotgun reads from metagenomic samples are converted into taxonomic profiles using a taxonomic profiler of choice. This gives the taxonomic tree with the relative abundance of each taxon. We then assign the branch lengths to the taxonomic tree and compute the pairwise UniFrac using the ISMB UniFrac algorithm, which computes the exact UniFrac in linear time. There are multiple ways of assigning branch lengths. It is ideal if it can be guided by additional biological data. In the absence of such data, we recommend a simple model of assigning branch lengths inversely proportional to the depth of the tree, as shown in the figure. We evaluated the performance of WGS UniFrac in comparison to the traditional 16S UniFrac under different settings by simulating data from two distinct environments with different within sample and between sample diversities, which were termed range and dissimilarity respectively, as illustrated in this figure. The performance of both WGS UniFrac and 16S UniFrac was evaluated based on the ability to cluster samples by environments. We further evaluated WGS UniFrac using real-world whole genome shotgun data and also compared our method to the OGU method, which is a recently developed method that measures beta diversity using WGS data. Let's now look at the results. Figure 1 shows the comparison between UniFrac compared 16S samples and WGS samples from simulated environments with different values of range and dissimilarity. The silhouette score was used to measure the quality of clustering, with 1 being the perfect clustering and 0 being completely random clustering. As we can see, similar trends are captured using the two data types. In Figure 2, the same experiment was repeated with different branch length assignment methods. From left to right, we have branch lengths increasing down the tree, all branches receiving the same length of 1, and branch lengths decreasing down the tree. Overall, we can see that WGS UniFrac is relatively insensitive to branch length assignments. However, among the three models presented here, decreasing branch lengths down the tree yields the best results. This is expected because it captures the general biological trend in real life that organisms at the lower part of the tree were more evolutionarily similar than those higher up. Figure 3 shows the performance of WGS UniFrac on real WGS data from different body sites. We observe that samples are clustered by body sites, which recapitulates the results of 16S UniFrac shown in literatures. In Figure 4, we can see that WGS UniFrac outperformed the OGO method in every setting. In conclusion, our study demonstrated that the UniFrac can be computed using only WGS data to offer reasonable insights in beta diversity. This shows that UniFrac is not strictly dependent on the presence of phylogenetic trees and can be applied to a much wider range of data. This concludes the presentation. Thank you for watching.